Chapter 3, Section 9, Principles of Respiration, Generating a Proton Motive Force. Okay, previous section, Section 8, we talked about the chaperones, the electron carriers. What they're delivering to is what is known as the electron transport. It's where those excited electrons are going to be passed on and their energy is going to be bled off. So NADH, FADH2 are going to deliver their electrons here as the hydrogens. That energy is going to be bled off, allowing for the transport of hydrogens across the membrane, either the mitochondrial cristae or the cytoplasmic membrane of prokaryotic cells. The passing on in this electron transport chain is going to take that excited electron and it's ultimately going to have it in a reduced state. It can then be passed on to oxygen. The final passing as to oxygen, hence the term final electron acceptor. And you look here. Here's NADH becomes NAD. Right here, FADH2 becomes FAD2. Electrons, their energy, as they're passed on to the different proteins, the flavor proteins, the dehydrogenase kicks it off here and it gets passed on, boom, 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 just as these get passed on, boom, boom, boom. As they're being passed, energy is being bled off, and that energy is being used to transport hydrogens. Finally, goes cytochrome C, cytochrome A, cytochrome A2 or 3, until finally it's now at a low energy state. Can pass on to our final electron acceptor, oxygen. Right here is the whole reason you have to breathe in. Hydrogens plus this excited electron and oxygen becomes water. If your oxygen levels drop, this stops. If this stops, this gets backed up. That gets backed up. This gets backed up. This gets backed up. NADH, FADH2 do not get oxidized. All of this shuts down. If this shuts down, those steps in the citric acid cycle where NAD and FAD got reduced, that whole citric acid cycle stops. Then you have to go and revert to the fermentation as we talked about 2 sections ago. So this whole idea of the electron transport chain is an exergonic process of bleeding energy off an excited electron so that you can pump protons, hydrogens, across the membrane. High proton concentration on one side, low on the other. You're setting up a differential. Okay? That's what this proton motive force is. You have an extremely high and extremely low, separated by a membrane. Cool. Complex 1, yeah, I'm not going to get into it too much. You can go and you can read who's the players there. I'm not going to. Complex 2. They call this an alternate entry because this is where FADH2 comes into play. Complex 3 transfers to QH2 ubiquinol to cytochrome C, pumps to hydrogens, blah, 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 blah. And then the final complex 4, which reduces O2 to water. This is where that final electron, okay, this is where it goes. More on the players. I am not going to ask you to individual players because this is not cell biology. Ultimately, by setting up that proton motive force, you are setting up the means of powering ATP synthase. N's and A's, so it's an enzyme. What prefaces it tells you what it does, ATP synthesis. It's the enzyme that's responsible for taking a diphosphate and adding it with an inorganic phosphate to form a triphosphate. In us, in aerobes and whatnot, things that are doing this with, powered by organic molecules, we refer to this as oxidative phosphorylation. Those organisms that are directly harnessing light, we call this photophosphorylation. The energy from photons is what's powering the pumping of the hydrogens to make the proton motive force, which will then power ATP synthase. ATP synthase is a membrane-bound enzyme. You have the membrane, the transmembrane components, the integral proteins here, holding this catalytic portion. All these subunits, alpha, betas. What they do is they spin. As they're spinning, they're taking a diphosphate, an inorganic phosphate, slamming them together in a chemical means to make ATP. This spinning is caused by the flowing of hydrogen on one side of the membrane through the base to the other side that has an extremely low concentration of hydrogens. Think about that. The proton motive force, you have the flow of hydrogens causing mechanical energy, the spinning of ATP synthase. The mechanical energy, the spinning, then joins two molecules together in a high energy state, chemical energy. Flow of hydrogens causes mechanical energy transforms to chemical energy. All because of excited electrons. You go through and you look at the different components, okay? F1, F0, the membrane, the the catalytic part that I told you about. Some form of this is found in nearly all organisms. This is highly conserved, so this is old, okay? This goes back to, common ancestor for all living things type old, okay? The ATP synthase in all of eukaryotes, homologous, not exactly the same. The ATP synthase in eukaryotes and prokaryotes, kind of sort of similar almost, but not completely, which means that goes way back. A lot of what we know about ATP synthase, it's because we studied it in E. coli. It's a little hard to study it, pull it apart, mutate the genes that make the parts of it if it's, a eukaryotic mitochondrial one. So we go and we study it in E. coli. Single cell, single circular chromosome. Can mutate it, go through the different parts, mutate the parts and see how that affects ATP synthesis. How does that affect the enzyme? How does that, affect output of ATP? And everything there is reversible. ATP synthase can go spin the other way. Utilize ATP, break it down to a diphosphate and inorganic phosphate, spin the other way, and then pump hydrogens itself. Reestablish the proton motive force. Problem is, that's a very high energy usage, and it's not very efficient. in generating the proton motive force. Now, for fermenters, okay, since they're not using citric acid cycle, they're not using the oxidative phosphorylation, so they're having trouble making, NADH, FADH, so they can pump to make that. Well, they'll use ATP synthase going in reverse to set up the proton motive force, reverse it, pump hydrogens back out, because remember in the last chapter when we were talking flagella and the arcella, that those were driven by the proton motive force, not ATP. Well, if they're in a fermenting state, it means they're going to have very little ATP, and they're not going to have the electrons to set up a proton motive force to make more ATP, so they got to use what they got. They got ATP, so reverse it, start swimming somewhere else where hopefully there's oxygen or whatever it is they need so they can get out of fermentation. So when you go and you compare lactic acid fermentation, here's our example. Glucose to 2-pyruvate to 2 lactates. In the process of doing this, glucose to pyruvate, 2 NADs, reduced to NADH, 2 ATPs, will yield, or there should be 2 ADPs, to 2A and 2 inorganic phosphates make 2 ATPs. Cool. That's substrate level phosphorylation. Problem is you're using up all your NADs and ADHs. So you have to regenerate. NADH, oxidize it, take those hydrogens, rearrange the pyruvates. You now end up with lactic acid. You now have 2 free NADs, which can go back. You go through glycolysis one more step. Whereas aerobic respiration, it's continuous glycolysis, citric acid cycle, electron transport chain, and ATP synthase, it's listed there behind my head. Look at the figure 321 in the book.